Okay, so hi everyone. Thank you for joining, our special session today. So we'll be focusing on discussing the use of four spatial technologies. But before I get onto the agenda for today, we have a very quick announcement from Gulan Liu. So thank you, Gulan. Do you want to proceed ahead with your announcement?
Thank you, Ajit. Hi, everyone. My name is Guolan. I'm a postdoc training with Dr. Gary Nolan at Stanford. It's so great to see all the spatial biology, spatial technology experts here. I just want to make a quick announcement that I'm co-editing a research topic on computational methods for spatial biology and immunology with two other colleagues. And so here is the right place to put the advertisement. So I'm going to put the information in the chat box. If anybody has any ideas, any thoughts that they want to publish, please feel free to contact us. We'll follow up with an email as well with the contact information. Yeah, please feel free to reach out to any of us, John Hickey, Christian Church, and the two other co-editors. Thank you so much for your time.
Awesome, thank you. So we will also send a follow-up email with all the relevant details. I know I've known John for a while, so I'm pretty sure that he'll do a good job in carefully handling your manuscripts.
Thank you so much.
So, all right, so for today's agenda, we are gonna talk about four different technologies. If I can get my screen shared. 1 second. I'm not sure big screen. Can you see my screen?
Mm-hmm.
Let me go on to full screen mode. Okay, there we go. All right, so, and you're seeing the right version, right?
Yeah.
There's too many screens to work with. Okay, so for today's agenda, we're going to have these four different technologies that's shown here. And as many of you might already know that, about a year ago, this association was established based off a conversation that we had during a cross-consortium junior investigator meeting. And one of the primary goals that we set out to achieve was to promote cross-pollination of methods, tools, ideas, and expertise among the community members. And for the last several months, we have had many leading figures in the field come talk to us about spatial technologies and how they're being applied to different biological scenarios. And we will continue to do that. But this event is something very special and our first attempt at really going back to those founding principles of building the community and getting the community talking to each other. So, which brings me actually to thank these amazing folks who have been behind putting all of this together. So yeah, so these are all the people who have been organizing all the events that you have been listening to in the last couple of months. So thank you very much. And for today's talk, when I was thinking about like what should be what should be the kind of the driving conversation that we should talk about when we talk about these different technologies. So I thought of three major areas that might be relevant when we initiate the conversation. So the first one being a conceptual thinking. Essentially, how do we leverage the uniqueness of each technology? Every technology has its own advantages and disadvantages. And what I mean by that is, for example, if you're looking at imaging data, sometimes we very often kind of force ourselves to treat an image data very similar to a single cell sequencing data, where you try to identify single cells and then look at expression profiles within them. By doing so, what's happening is we are losing a lot of information with regards to morphology or the marker distribution within the cell or how the cells are oriented and so on and so forth. So how do we capitalize on each technology and the kind of changing the thinking about how data is derived from all of these different technologies. And obviously the practical implementation, that's the nitty-gritty of the technical aspects with different technologies, and how do we communicate that, how do we set standards, and how do we work with reproducibly getting the same results over different methodologies. And finally, of course, exchange of methods, tools, and ideas for interpretation of the data. This might be a bit of a stretch for the last point, which we might have to go on to a different meeting. But we can definitely talk about many of those things. So we have 4 different technologies, as I talked about previously. The first one being CYCIF, and then CODEX, which are both spatial proteomics, which are imaging based. So we have with us Jia-Ren, who is the inventor of CYCIF and who I get to work with every day. And then we have CODEX. We have invited Domenic and Huan, who both work at the Broad Institute. So they work with Sammy, who many of you might already know. He's a group leader leading the imaging platform in the Broad Institute. And then for spatial transcriptomics, which is sequencing based, we thought that we will initially discuss about SlideSeq and Omics. Obviously, there are other methods which we will potentially go into on a later meeting. So for SlideSeq, we have invited J.B. Marshall, who was again a group leader in the Broad Institute and has recently moved to Solid Biosciences. And for Geomics, we have a presentation from Angela Cruz from Vanderbilt University. And I've also invited Roxy, who runs most of our Geomics experiments at the Harvard Medical School. So with that, I'm gonna pass on the mic to Jerry. And since this is mostly going to be a discussion-based session, if you're comfortable, obviously turn on your videos, and that will help facilitate the discussion.
Thanks, Ajay. Thank you so much to invite me, and yeah, great opportunity here to meet everyone. Probably my first time here. So I would take the liberty of, since I am the first speaker here, so I will be more general but less specific to the CYCIF itself, because I think, yeah, based on what Ajay said, it's more a discussion to how to join everything together, but not necessarily tied to a specific technology, right? And even start with maybe even more general concept, because I think everyone here might have know and use, or it's your daily routine to use single cell sequence. And just get a review from Nature Protocol here to show you that's how the single cell sequence developed over the last decade. And it's a lot of improvement, as well as now you can see it's getting, I would say, bigger and bigger. Yeah, bigger always better. And in a way, it's not trivial, because you need to accumulate to a necessary critical mass in order to get understanding of how the biology behind. And if you only can sample a small amount of the cell, it won't tell you too much. And I draw the line here is few of the early method to use for the spatial omic approach, right? For example, like MixIF, that probably way before the GE method. That's how basically our precedent or our commercial solution for CYCIF. And also, most of you may know the INC. That's probably, so you can see we have lag behind of single cell sequence, maybe 5 to 7 years. So it's about the time for spatial omic technology to take off, I would say. And Ajay already did AI, and it's probably what we will try to cover today, right? We have several different aspects or several different approach you can do spatial omic. Either on here, we kind of separate between the spatial, although some people don't like that name, but spatial proteomic approach, something like CYCIF, CODEX, OPRA, or some other similar method, as well as spatial transcriptomics approach. Either is Murphy's RNA-seq. Here is divided by, I think you probably can now really appreciate, it's either divided by it's a sequence-based or in situ-based or image-based. So they all, like Ajay just mentioned, they all have their strengths and weakness and uniqueness. And as far as we can tell, I think that I will go back to tie back to the to the image method or CYCIF, or it's more general like image-based or antibody-based spatial proteomic method. They have their limitation here because we use antibodies. So the number of markers is limited, unlike the some sequence approach or even the in situ approach, you can reach more higher biomarker you can detect. But the other aspect is less being considered is because all this kind of the image approach, we can push the resolution as high as possible. So we will have subcellular resolution as well as like a morphological resolution. This resolution can be multi-scale. We can see as small as micronuclei inside the cell, as big as the whole tumor. So in a way, I think that's something very different from the spatial omics, as well as we, in a way that spatial proteomic approach can process, the amount of sample can be processed much larger. So in a way, in one single experiment, you can process 50, 100, or even in the future, maybe even more. So that's something maybe take in mind. So, but in last point, I don't know if that's interesting to you as well, but for us, we also try to think about this a lot because in a way, although we only can image 30 to 60 markers, like in general, but in each marker, in each cell, we don't have the problem in terms of drop off. So you can have a continuous distribution of your data. And for the sequenced approach or in situ approach, they are a different kind of the measurement right there. So in a way, try to come up with the approach like specific to proteomics, or try to come up with an approach that can join this together is still underdeveloped, I always say, because I think in a way the spatial transcriptomics in the methodology go much faster than spatial or than the image method. Sorry, that's kind of a long introduction for everything, but now we tie back to CYCIF. And CYCIF, I think maybe some of you heard about that, but it's similar to what you do with CODEX, right? So if that, I think now CODEX become a more popular approach, but the same idea, you can do this kind of cyclic staining. So in each cycle, you only stain a given amount of marker. In here, we use 3 markers in one cycle. And then you can erase the signal in any mean. For CODEX, they use the specific DNA conjugate antibody to do that. For us, we're doing this very simple chemistry to using hydrogen peroxide to bridge the fluorophore. So we can repeat this procedure multiple times. And in the end, you get this large image mosaic that assemble together and go through the pipeline. I think there are a lot of developers in terms of the computational approach, image processing approach to get your final data. So the image, it look, so for me, myself, I like this kind of approach, although it's because we've been working on that for years, but also in a way you can have a direct appreciation of your data look like, right? You see this image, you understand your data, but it's less, it's more convoluted if you do a transcription approach. So I will start right here. In a way, it's more a perspective and more a discussion point for the other methods as well. So in a way, we are thinking we can translate this image to a data, but it's not necessarily just single cell data, right? So because once we image assemble, we have all the structure, all the spatial information. So we can assemble a different layer of data. So from the very beginning, the intensity of single cell, the gated markers, all that stuff, then you can come down with the cluster cell type. All this is on the single cell level. That becomes very useful information for say. And that's been actually much more likely to homogenize or communicate with other modality. But for the spatial omic or spatial proteomic approach or image-based approach, we can go one step further, or usually we will go is one step further to try to see if we can see any kind of the cell-to-cell communication, right? So cell-cell interaction, as well as some sort of special topic or special motif, you would say, then that would be a building block toward even higher structure, I think that's even less being approached. It's like this kind of domain or landscape that's assembled by this small building block. And that actually, if you think the other way around, a more traditional way people look at this tissue is histopathology. They always look almost, we would say, this kind of top-down approach to try to look at the large morphology, then go to the single cell. So in a way, we can try to see if we can come up with this approach that takes into account of all there and try to join them together. I think, and also in a way, also join to the other modality. I think that's kind of what I was thinking right now, the future of all this spatial omics approach. Sorry, it's a little bit deviated from the topic we have discussed, but hopefully it's still some useful information. And I will stop right here.
Great. Thank you, Jerry, very much for the introduction. So yeah, we do want to make sure that we have enough time to also discuss and bring up topics that we had. Even if you don't feel like you were able to bring up everything you want to talk about in this session, please type it in the chat and we'll collect everything. And then we can still follow up on that. So I know we had some topics regarding CYCIF in the discussion sign-up sheet that we had. So I don't know if anyone wants to bring up their questions there. Is there anyone like working with CYCIF? What are common issues, challenges that you face with that technology? Ajit.
I know that Charlie had a question. If I saw Charlie's on call, do you want to bring it up, or I can read out the question for you as well? So probably I just read the question and Charlie can jump in at any point. So he had asked, like, there are various antigen retrieval methods and how it impacts antigen retrieval efficiency.
Okay, yeah, I think this, although this question is for CYCIF, but it's a more general question for any antibody-based approach, I would say. So we've been experimenting this for quite a while. In a way, if you're thinking about CYCIF or CODEX work, in some way we need to do it retrospectively. So because if we do in a traditional IF, let's just say traditional IF or IHC, we can optimize this for individual antibody. That's much easier to do. But for CYCIF or IHC or the CODEX or whatever you think about this kind of method, because every antibody will be used simultaneously, right? So if we are optimized for one, it might be not optimal for the other, right? So in a way, we try to do here for CYCIF, at least for CYCIF, I don't know for sure for the other method, but when we do our antibody validation, we will use a constant protocol to do all the antibody. So in a way, we select the antibody that fit our antigen retrieval. So in a way, it's not necessarily answer your question, but that's what we do here. So the antibody we've been used, it's probably only optimized for the condition we are tied up to. To say that we might be able to tweak that for a specific purpose, but like you're aware of, once you change that, a lot of antibody will validate before might not work or might not work as well. So that's need to be re-optimized or at least retest again for all this. And recently we actually have another finding, even as like subtle as the blocking solution you're using, we also found they have a profound effect on the antibody standing signal we see in our system. And we probably think this will be general for the other system as well, because antibody is antibody. So in a way that's, it's just, I want to say, they need to have a more explicit description about the protocol, as well as how the antibody validation been done. Then people can use that information to select the antibody they want to use in their workflow. Does that make sense?
Yeah.
Yep.
Okay. Yeah. So just a very general question, what kind of equipment do you need? Do you need special equipment if you want to start CYCIF?
No, I would say no. But we start with CYCIF with a very, very small instrument, very cheap instrument. I just say from GE, surprisingly from GE. Although the one thing you think about CYCIF, although we have less and less constraint right now, in the early day when we do CYCIF, the main constraint we have, because you need to cycle this, right? Every slide need to put on the same image or same microscope for many times, and they need to find the same region or same position every time. So the only requirement here is your machine or your microscope need to be able to do that. Yeah, you just need to can go back. If next time you put on the same slide, you just need to go back the same position. But for now, because our improvement on the computational process workflow in terms of register multiple cycle, I think we have less and less constraint on that. You can allow a large movement of your tissue, but still get the registration work. So that's been solved a little bit by the computational approach. The other thing is sensitivity. So in a way, because we using only direct fluorescence conjugate antibody, so our signal to noise ratio is not as great as the other method that has amplification. They can make a signal to noise bigger. So your microscope need to be sensitive enough to pick up this signal. But if you use a little bit more than fluorescence microscope with a reasonable CMOS, you should be able to achieve that signal sensitivity. Mm-hmm. But we do have some issue before to see some other people use like microscope still using like OCCD, and they just cannot see signal because they just don't have enough sensitivity to pick up that. Mm-hmm. But I think that's a good thing or bad thing in a way. If you don't need amplification, the first thing is you can have less step, right? It's more straightforward, mm-hmm. as well as the signal will be more proportional to the target you try to see. So in a way that let's still keep the dynamic range mm-hmm. there. Okay. You want to measure.
Yeah. Thank you very much, Jerry. No I think problem. In the interest of time, we need to move on to the next topic. But
one last thing. Can I have one last clarification?
Last sentence.
Yes. I actually not an inventor of science. I don't like that title. And the only thing I invent is a name. All these methods is built on the previous experience of this kind of cyclic procedure. And we happen to optimize that and share with the people. So just want to say that.
Okay. Very humble. Yeah. Of course. Yeah. If you have any other, like if you can still continue to answer questions, if there's anything in the chat, that would be great. But I think we would like to continue to the next topic, which is still in the same area of CYCIF. So it's going to be CODEX. And yeah, if you could, I think we have Domenic and Huan here to give just a brief introduction about the technology. That would be great. And then we can start discussing a little bit of the challenges. So please go on.
Okay. All right. Can everybody see my slides?
Yes.
Okay. First, I want to thank you all for giving us the opportunity to present here on our experiences with CODEX, and hopefully we can have a good discussion and work on some improvements and let you all know if CODEX is the right thing for you. So the way that our slides are structured is we highlighted some of the considerations and kind of obstacles that you may encounter when running CODEX. So the first part that I'll talk about is imaging experimental considerations. So one big point with multiplexed imaging is throughput. Both CYCIF and CODEX are relatively high throughput, but with something like CODEX, there are some limiting factors. For example, with the commercial setup that you get, when you contract with Akoya, you get the CODEX instrument here. This is the beta version that we used to use, and it's fitted for a wide field microscope. And wide field microscopes are great. The Zeiss is a good microscope, but you may not have as nice of pictures, and you may get some out of focus light. And you will have to do something like acquire Z-stacks, which will, you know, multiply your data exponentially. And then we have considerations like how many colors you can image in, the standard 3 colors usually on the wide field, and then the imaging time. We have been able at the Broad to build a custom setup with our Dragonfly spinning disk and focal, and fitting the CODEX instrument to it, along with some Python code, so that we can acquire single plane, just stabilized images. No need for Z-stacks, everything stays in focus, and because we're using confocal, we're only getting the in focus light getting to the camera. And with this, we're able to cut down, say for a 10 cycle CODEX run that used to take, you know, something like 20 hours on the wide field microscope for a much smaller field of view at 20x. We can now acquire an entire 18 millimeter by 18 millimeter cover slip. That's the imageable area of a 22 by 22 millimeter cover slip in about 8 hours. So that's an entire cover slip with up to about 20 markers for 20 or 30 markers for an experiment this size in about 8 hours. So we've been able to improve greatly on throughput. And the code that is running this is actually on our protocols.io and can be modified to run with any microscope. That's not what Akoya currently supports. From hardware, we can also move on to more wet lab focused with throughput, and that's tissue formatting. Most of us are familiar with whole slide imaging, or if we're thinking more clinically with pathologists, and one thing you can get from patients are core needle biopsies. And what we also have are in the same realm, core needle biopsies and whole tissue, the best of both worlds, which is a tissue microarray where we can fit, you know, on this core, this is 120 cancer types on a single 22 by 22 cover slip. So tissue formatting your sample is very important for whatever information you want to get. If spatial context is really important to you and you have a lower amount of samples, maybe a whole tissue is best for you. But if you want to find a good balance between spatial context and throughput, it might be easier to design a TMA and core your samples with the regions that you would need to focus on. And then finally, for experimental, I can just talk quickly about antibodies and fixation. Something that Jerry mentioned before was something like antigen retrieval and blocking can greatly affect our staining, especially for something when you're designing your own custom antibodies. Akoya has a great catalog of antibodies, and there's a community source list. But for certain projects like this project that we were working on actually with Jamie for Kidney Atlas, we can see that the custom conjugations, you know, when they work really nicely. But when they don't work, it's kind of hard to tell what could have gone wrong. Was it the antigen retrieval? Was it the clone? Most of the time we keep our antigen retrieval conditions the same, and we cycle through antibodies that then fit our conditions, much like Jerry said. And finally, for another experimental consideration for the tissue is fixation. So this is just a quick comparison between fresh frozen and FFPE, two most common types. And what we noticed and what we like to do with our samples, we like to work mostly within FFPE because we find that FFPE preserves morphological structures better than fresh frozen tissue. And then from there, I'll pass it over to Huan.
Hi, this is Huan. So once we acquire images with a microscope, we're gonna need to transform the raw data into a format we can visualize, explore, and create reports on, as well as a tabular data format that enables downstream analysis. So to do that, we integrate our own CODEX image pipeline, including computing engine setup, data preparation, stitching, and registration using MC Micro, developed by Peter Sauger's lab, leading by Dennis Shapiro, and segmentation using Mesmer or Omics, and cell type assignment using Aster or Celesta, which is I'm just trying to test recently. With this pipeline, we have processed about 30 tissue samples, with a big part of that is for H10 project. As you can see, next slide, please. So here is an example of pipeline improvement on segmentation, as segmentation is critical for extracting single cell data. Experimentally, we tried different markers, and this Lamin B1 marker can clearly show the nuclear envelope with super clean signal. And this sodium potassium ATPase marker did a pretty good job on marking the membrane. So by using these markers, the segmentation result was improved. And also we keep track of the papers and adapt the state-of-the-art method, Mesmer. As you can see, both the whole cell and the nuclear segmentation was done pretty accurately. Next slide, please. So once we have single cell data derived from the multiplex imaging, we are analysis ready. Here I'm showing some standard analysis we can do. On the left, that's cell type assignment with a bar chart to show the accuracy. And we're comparing the results from our data to the Aster paper, and it's comparable, if not better. And there's some other figures I will not go over, but this is what we're doing, and we're also working with pathologists to better formulate what question we're trying to answer with the data to fulfill the research goal. And with that, and also one more thing is for other like visualization was done using open source packages, and also we also use Minerva Author and Story to do interactive data exploration, which is pretty cool, and worth another demo in the future if we have time. But with that, I'll stop here. Thank you.
Thanks a lot. Yeah, that was really informative. We have some questions collected. I will start with the first one, a quick one on the experimental side. So I'm actually also a CODEX user, and one issue that I'm having is, so I work with tissue microarrays, but then we use a keyence attached to the CODEX setup. And as you introduced, Domenic, in the keyence software, or the CODEX software, basically gives you a single Z-plane or a Z-stack that you can set up. But then in TMAs, very often I feel that you don't get all the different cores in the same Z-stack, or if you do, you have to make a huge Z-stack, which takes enormous amount of time. So I wonder, like, is that something that could be fixed with the software, or what do other users do? Does anyone have solutions for that?
So I will say that we found that our solution was fitted mostly to our setup. So we built the Dragonfly setup to address that issue of trying to get high quality data, but with actual less data from those Z-stacks. So the way that we acquire our images is that we use an Nikon Perfect Focus, which sets an offset from the objective to the cover slip. And that offset is kept across the entire slide. You can think of it like a better autofocus, but you're storing a single value, and then it tracks across the entire cover slip. And we've had pretty nice success with it as of late. It took us a while to get it implemented, but we very rarely have out of focus areas of our tissue. So with a wide field microscope, I would say you're probably still stuck to acquiring those Z-stacks, especially with TMAs, maybe to build the cores closer together, so you're not having as much of a shift across. Like you can, like, if you put the cores closer together, you can correct for if the slider cover slip itself is not completely flat or any chromatic aberrations there.
Mm-hmm. Sounds good. Okay, thank you very much. Then there's a question in the chat from Anina. If you want, feel free to also unmute yourself and ask your question.
Okay, hi, thanks for the presentation. I was super interested to see that you are using some combination of markers for the kind of cell segmentation or cytoplasmic markers. Could you a little bit explain what you did there?
Sure. So we tried different ones. So for mesmer, they required two marker. One's like a nuclear marker, one's like a membrane marker, and we were using lamine B1 for nuclear and sodium potassium for the membrane. And you can also try the DAPI, but I think the lamine B1 works better. And also I saw a second question, like what's the size range of the stitched image? For the one I just showed, it's like a big TMA sample, and it's about 250 gigabyte for the stitched image.
Sorry, one more. So about the cytoplasmic markers, not the nuclear membrane, but cytoplasms.
I will have Domenic answer that question.
Yeah, so we actually settled on the NAK2PAs off of what we saw with MIBI. And we saw that was a very nice general membrane marker in MIBI. So we thought it would translate really easily over to CODEX if we just conjugated it correctly. And what we've seen is it stains, every membrane that we can get. We do have some issues though with areas of higher density tissue. It doesn't fully penetrate, and therefore we don't have as nice of segmentation from the membranes in those areas. But generally, it works pretty well.
And the protein is a protein?
Yes. Mm-hmm.
So which, I'm sorry, I missed it. Which protein? Can you
maybe sodium potassium ATPase.
Oh, okay, I see. Yes.
Yeah.
Okay, perfect. Thanks.
Mm-hmm.
All right. We also had a question on the sign-up sheet from John Hickey, who couldn't be there here today, unfortunately, but I think he brought up a really interesting and issue that I've also thought about sometimes. So the topic about batch correction algorithms, because at least in every run, every experiment, every machine may have some batch effect. So it's not super straightforward to compare different samples, different CODEX runs that may also be true for CYCIF. So he was basically wondering if anyone is looking into algorithms to tackle these batch effects, or if not, if people are interested to explore opportunities to do that more. We could, he suggested even a hackathon or something. That would be, yeah, let's just open it to the discussion. I don't know, hey, Dean, if you want to speak on that, or do you have another question?
No, it was actually on this. So we've been talking about batch effects and CODEX. I'm computational, but I'm collaborating with a group here that have actually got a lot of experience in CODEX. They've been generating images for about 2 or 3 years, maybe 2 years. But yes, we see batch effects. The primary batch effect that we're dealing with is where there were different pre-processing of some of the tissue. And we were talking with Akoya, and we're thinking of setting up a benchmarking TMA. We've got a lot of tonsil data. And what we were thinking of doing was using that's been processed in different ways, putting it on a TMA and basically creating it as a benchmarking, benchmarking tool. Now, we haven't got that far along in that process yet, but it was what we were thinking, what I was saying we could try and do, and then make it available to the community as a potential benchmarking resource. So if other people are interested in maybe generating a couple of different benchmarking studies that we could actually come together and do something like that, that's something I'd be happy to get involved with.
Yeah, that sounds great. Jerry, do you want to add to that?
Yeah, sorry. Yeah, because we do have a little bit more experience here since we run in CYCIF for over maybe 7 years now. So a lot of this effect, like I think just mentioned, is actually, we call it pre-analytical, because that's determined by how you fix this tissue, how you process this tissue, all that. And that's not necessarily tied to battery effect, because battery effect is different, but like a sample to sample variation, it's quite challenging in the tissue image. In a way, we try to generally deal with that is using the downstream analysis to deal with that, right? So almost like, I would say, almost like you run a flow cytometry experiment or cytop experiment. There are a lot of ways to do, we call the gating. So basically you try to determine which signal range will be positive and negative. Then you use that value to do your downstream analysis. So in a way, I would say currently we don't feel that the absolute value that's coming from the image analysis will be straightforward to use to your downstream analysis. So in a way that some form of human intervention or some algorithm to do this kind of gating would be necessary to do that. And once it's been gated, then you basically working in the same standard. But in a way, it also loses a lot of information. So I totally agree that that might be an open question here to see if we can find a solution, general solution to do normalization.
I feel like that is actually a topic I hear gains a lot of interest, and a lot of people face the same issue. So as an idea, maybe we can try and find another session or team up and kind of troubleshoot on that specific area a little bit more with all people involved working in this. But we can do that for another time. I think so
if we want to do this, we need to have like benchmarking slides that we can use where there's replicates, where you know what we know the condition is, and you can basically design. If that's available, I work a lot with like bioconductor and statisticians who've done a lot of this before. I'm in bioconductor. I'm happy to put this available to people, or even bring this up at like the annual meetings and different things like that, so we can like start kind of a bigger process on this if we want.
Yeah, no, that sounds great. I think we can follow up via email and see if we get a team together who's interested in tackling this a little bit more. So we'll follow up, but I want to get to our next speaker, who's Jamie Marshall, and she will introduce SlideSeq a little bit. I want to give you a heads up that we will not cover GeoMX today. So Jamie, you will be the last topic for today, and we'll keep the GeoMX for another time. But if you had any questions that you were eager to get discussed today on GeoMX, please put them in the chat and we'll note them down and discuss next time. But now, Jamie, thank you very much for introducing SlideSeq.
Yeah, no problem, happy to. So I have kind of less in-depth slides, but I'll go through how the method works. So the first step is to have a SlideSeq array, which is shown here on the left. And arrays are made on a glass cover slip with liquid tape, in which you then centrifuge the beads down onto the liquid tape, and that's how they stick. The beads have a PCR handle, a spatial bead barcode, a UME, and then a poly-T, which binds to polyadenylated RNA. And so sequencing these barcodes on each of the beads is actually what gives us the spatial locations. And this is done in a flow cell shown here in the center, and it occurs right now on a confocal microscope, but it can also be performed on other microscopes, and it takes about 2.5 nights. And just to show you some of the raw data from this, each window is one small area of an array in which we're sequencing in each pane the 14 different barcodes on the spatial bead barcode. And then underneath is the computational predictions. And this is what gives you the bead barcode for every single position. Once you have the beads sequenced and their spatial locations known, you're then ready to put tissue on. And so for me, I was using kidney tissue, and I took a 10 micron slice of kidney tissue, mounted it onto the SlideSeq array. You then transfer this, basically the glass cover slip with the tissue on the beads, into a tube, into SSC buffer, and allow the RNA to bind to the beads for about 15 minutes. You then perform reverse transcription, second strand synthesis, and tissue digestion. So now the beads are in suspension, and you amplify the cDNA. You then perform normal library prep steps in which you prepare the cDNA for sequencing, similar to single-cell RNA-seq. And we do sequencing on a traditional Illumina sequencer. And the output is a little bit different. So you'll have Illumina barcodes and a DNA count matrix, and that's what you'd normally get with single-cell RNA-seq. But then we map on our in situ sequencing spatial bead barcodes. And so you have a count matrix that has spatially resolved Illumina barcodes. And that's ready for processing. I didn't really go through all of the data analysis steps, but I can just direct you to my paper if you're interested in that, but I just thought I would quickly show you what some of the arrays look like. So this is arrays from mouse kidneys, cortex enriched areas on the top and medulla on the bottom. And then similarly for human kidneys, cortex on the top and medulla on the bottom. And I can just tell you without getting into it, that we're able to see all cell types in the regions of interest, and then did further characterization. So I'll go ahead and stop here and answer any questions.
Thank you very much, Jamie. Do we have any questions from the audience? Or
I have one question. Jamie, how do you deal with, like, since you have like 10 microns, even with the 5 micron section, there's the problem of like cell overlapping with each other. How do you deal with that in 10 microns?
So the RNA basically on the interface of the beads is the only RNA that's really going to bind. So you're not going to get like RNA binding or being able to reach the beads from across the 10 microns, if that's kind of your question. A similar question would be, so I should have mentioned this, the beads are 10 microns themselves in size. And so that will capture between 1 to 3 cells of information. So Visium is larger, it would capture 10 cells, it's 100 microns. But with 1 to 3 cells, it is actually kind of a computational challenge to actually map cell types, and you're not going to have perfect mapping, as you can imagine. But Seurat has a nice method for label transfer where you can use information from single-cell RNA-seq to attach labels. And that's what we used and find work the best. And then when you're doing like clustering and differential gene expression, you just have to remember that you could be capturing more than one cell. So you might have to like tighten your edges for the subsets that you're analyzing, or just dig through the genes that come out of the differential gene expression and make sure that they're actually would be expressed in the cell type you're interested in.
Okay. That makes sense. So one of the main challenges, or one of the main reasons people like spatial transcriptomics, or comparatively would prefer spatial transcriptomics to the current spatial proteomics method, is that you have a larger dimension of data. But if identifying cell types in spatial proteomics is not that challenging, but identifying cell states and pathways that are activated within them is likely very biologically interesting. Yeah, I guess David has a question. Go on.
Yeah, I have a pretty simple question. I was wondering if you have seen any, if you've tried out any fixation strategies or permeabilization strategies, like what is used in Visium. From what I understand, SlideSeq has no permeabilization, which is probably why, like you said, the only RNA that you capture is that which is at the interface between the beads and the section. But
so I personally, like, because I helped develop SlideSeq version 2, tried out permeabilization. What I will say happened is the image. So if you looked at like the same tissue section on different arrays, testing out different permeabilization, it became less clear. So you had kind of more fluidity in the structures and kind of spreading across the beads. So that's why we decided to go without any permeabilization. I know other people, I'm not currently in the group anymore, so I don't really know like what's going on actively right at this moment. But in the past, they've tried out fixation and couldn't really get anything to work. So I think it's still kind of in development, and obviously they're working on the next best technology. But what I will say, and I'm very excited for like my own role now in the company that I'm in, is Visium is coming out with a 5 micron bead product. I thought it was supposed to be out this year, and I'm very disappointed because I heard it's early next year now. That should be like really cool because it would actually be one single cell resolution. Visium has, I mean, they have 100 micron beads, but they tend to have like higher capture of RNA and more in depth than SlideSeq currently does. So it should be a really good product. Hopefully, I mean, I'm expecting it to be expensive, but we'll see. But I want to use it in my current role. So I'm kind of waiting for that.
Yeah, I think that product has been pushed back 4 times now, and the prices that I've seen are pretty astronomical. But sorry to come back to SlideSeq, it sounds like the with fixation and permeabilization, the issue that you've seen or that others have seen is just diffusion issues.
Yeah.
Okay, cool. Thanks.
So with the fixation, like not being able to capture RNA, and then with the permeabilization, definitely diffusion.
Yeah, and I guess by fixation, you're probably referring to some sort of cross-linking, like PFA, like okay. Yeah, exactly. Methanol fixation would be a whole different ballgame, but yeah, cool. Thank you.
Yeah, you're welcome.
Okay.
Sorry, yeah, it's just maybe for Jamie. Thanks for the presentation. It's more of a physiological question also for myself as well, because since we have VCN, right? So when for people here as well, when do you want to choose more like a commercial, ready to use product versus this more like a homebrew things? For us, I think we have our answer here, but I just wanted to listen to you or everyone's opinion why you choose one versus the other. Maybe that's an interesting thing to learn. Yes.
So for me, I wanted the highest resolution because I was studying kidney. I'm in muscle now, but I was studying kidney before, and there's very small structures that are really densely packed. So with Visium, it would be really hard to study anything at any physiological level, especially like when podocytes could be the only cell type that's affected in a very small structure. I will say SlideSeq is definitely not easy to use. It's easier for me because I'm like in the group and kind of help develop it. But if you're a first-time user, getting SlideSeq to work is probably going to be really difficult. And plus you have to get the arrays shipped from someone who's like able to sequence them, and that's also difficult. So I think if you're a first-time user interested in problems that you could answer with like less resolution, Visium's definitely the way to go, because the computational processing is also more streamlined. 10x has a really nice platform setup. Once they release the 5 micron product, that's definitely the product I will be using because it's the highest resolution and will have 10x's really nice ease of use in the wet lab as well as the computational platform. So yeah, I think it's just a mixture of the two. And then as Ajit was saying earlier, proteomics is also really interesting. I kind of think you want to use, depending on what questions you're asking, a combination of all of the methods, right? Currently, because you want the high resolution spatial transcriptomics, but you don't get as in-depth sequencing. So you may want Visium to pair with that, or single cell at least. And then proteomics is also really powerful because a lot of things that we see in transcriptome don't actually translate to what you're seeing at the protein level. And so you do have a lot of work to make your transcriptomics findings actually validated and kind of move on to real biology. And then for me at a company, we're trying to translate this into therapies for patients. And so it's just another jump that you have to make. But I don't know, for the people doing the proteomics solutions, they're not that easy to do like outside of labs that do them as well. So like for me at a company, I wouldn't have access to any of those. So it's really difficult, but interesting, and hopefully it will get better, as things progress and more technologies emerge.
Thank you.
Yeah, I agree that it's mainly ease of access, I think, for people who are like more, not so much on the technology development, but rather after answering specific biological questions, you tend to rather go for the commercial assays just because you can get it, you can order it. Unless you have maybe local collaboration that gives you this access and the expertise, it's just what Jamie mentioned, basically the ease of access and all of that. And yeah, well, thank you so much, everyone, for the nice presentations. This was really great, and I think we can probably even discuss much longer on that. Thanks everyone for discussing. Please send us any questions if you had anything else coming up. We'll make sure, and also suggestions for future sessions, we'll make sure to implement that and basically tailor everything to your needs. Jit, do you have any final notes to add to that?
Yes, thanks to everyone who joined us today. Thank you. See you.
All right. Thank you. Bye-bye.
Bye-bye.